Cell Respiration by Nikki Ella.
The energy derived from the breakdown of glucose is used to synthesize ATP from ADP and inorganic phosphate, and the overall equation for cellular respiration is C6H12O6 plus 6O2 to form 6CO2 plus 6H2O and 36 ATP.
The first two steps of cell respiration are glycolysis and pyruvate oxidation. And the second two steps are the Krebs cycle and the electron transport chain. Glycolysis! Glycolysis is the first step in cellular respiration. It is the process of gathering energy from one glucose molecule. This glucose is broken down into two pyruvate molecules, and the process occurs in the cytoplasm of a cell. The first step of glycolysis is that energy is made available when two ATP molecules are used. This allows energy to be gathered for the process to begin. Then, one of the two ATPs attaches to a phosphate group. This phosphate group is at the end of one of the glucose molecules, and this creates glucose 6-phosphate. Glucose 6-phosphate then rearranges to form its isomer of fructose 6-phosphate. This formation makes fructose 1,6-biphosphate. The backbone of the glucose molecule then splits to form G3P and DHAP. Now that there are two G3P molecules, each one gives up one hydrogen and two electrons to NAD. NAD is then reduced to NADH, and the G3P that binds with an inorganic phosphate. This combination forms a very unstable molecule called 1,3-BPG. To form ATP, 1,3-BPG gives its phosphate to ADT. From this, 3-PG is made. This step produces 2 ATP from a process that is called substrate phosphorylation. 3-PG then arranges to form its isomer of 2-phosphoglycerate. This isomer then gives up one hydrogen and one oxygen to form water. This then forms PEP. Since PEP is very unstable, it breaks down and forms a molecule of ATP, before it is able to release enough energy to transfer a phosphate to ADP. In summary, glycolysis is able to produce 2 NADH, 2 ATP, and 2 pyruvate. Pyruvate oxidation! Pyruvate oxidation is the second stage of cellular respiration and occurs in the mitochondria. To begin, a pyruvate molecule enters the matrix of the mitochondria. It is able to do this with the help of a membrane carrier. Since there is an unwanted carboxyl group left over, it is released as waste in the form of CO2. The coenzyme A then attaches to the acetyl group that is part of the pyruvate. This molecule is then oxidized and NAD is transformed into NADH. This produces an acetyl-CoA. The acetyl-CoA next enters the Krebs cycle to continue respiration when energy is needed. In summary, pyruvate oxidation produces 2 NADH molecules. Krebs cycle!
Krebs cycle is the third stage of cellular respiration and is also known as the citric acid cycle. The Krebs cycle occurs in the matrix of the mitochondrion, and the first step is acetyl-CoA reacts with oxaloacetate to form citrate by removing the CoA. Citrate undergoes isomerization to become isocitrate. Through a redox reaction, isocitrate becomes alpha-ketoglutarate by oxidation, and NAD+ is reduced to form NADH. Alpha-ketoglutarate forms succinyl-CoA by binding with a CoA and is oxidized by NAD+. CO2 is released. Succinyl-CoA loses its CoA to become succinate. Phosphate from the matrix displaces CoA, guanosine disphosphate, also known as GDP, to become GTP, and ADP forms ATP. FAD oxidizes succinate to form FADH2, and succinate becomes fumarate. Water is added to fumarate to become malate. Malate undergoes a redox reaction with NAD+ to produce NADH and oxaloacetate. In summary, the Krebs cycle produces 6 NADH, 2 ATP, and 2 FADH2 for each glucose molecule.
Electron transport chain!
The electron transport chain, or ETC, is the final step in cellular respiration and takes place in the inner mitochondrial membrane. It creates ATP through oxidative phosphorylation, and there are 5 major components. The first complex is NADH dehydrogenase, followed by succinate dehydrogenase, cytochrome BC1 complex, cytochrome oxidase, and oxygen. The proteins are in order of weak to high electronegativity. In the first step, NADH binds to NADH dehydrogenase. FADH2 binds to succinate dehydrogenase, and the hydrogen ion enters the intermembrane space. Ubiquinone, or UQ, carries electrons from the NADH dehydrogenase to the cytochrome BC1 complex. The cytochrome C then carries electrons from the BC1 complex to the cytochrome oxidase. FADH2 starts at the succinate complex. 2 electrons leave cytochrome oxidase and combine with oxygen and hydrogen to form H2O. Oxygen is increased in electronegativity to strip of the electrons. Overall, it creates an electrochemical gradient for ATP synthesis. In summary, the electron transport chain uses NADH and 2 FADH to produce ATP and release waste.
Chemiosmosis!
Chemiosmosis is a process in which ATP is synthesized using the energy of an electrochemical gradient in the ATP synthase enzyme. It occurs after electron transport chain, and ATP is then made through oxidative phosphorylation.
Yay!